Okay, we're going to talk about genetics using Punnett squares. So we all remember that early genetics was a study which began with Gregor Mendel, and Mendel noticed after he was observing pea plants that they had flowers that were either violet or white, or let's say that they were either tall or short, or so on or so forth. And then he began to study how those traits segregated when the new pea plants were born. Okay, he worked almost 150 years ago, and nobody knew about genes or DNA or just that even any of that existed. So here are the traits. I told you there were a bunch that they observed. They were seed shape. We talked about round and wrinkled. Seed color, yellow and green. Flower color, there's purple and white. So remember that genes carry information that determine the traits. Traits are those characteristics that you inherit from your parents. And then genes are located on chromosomes, which I'm hoping this is all very much review for you. Chromosomes come in pairs, and there are thousands of genes on one chromosome. So remember, they come in pairs. That's because you have one copy of a chromosome from your mom and then the second copy from your dad. So in a human cell, a cell's nucleus contains 46 chromosomes, or 23 pairs of chromosomes, meaning this chromosome right here might be from mom and this one might be from dad, but they're both copies of chromosome 1. They have the same kind of genes on them. Chromosome 2, copy from mom, chromosome 2, copy from dad. Again, they have maybe different alleles of that genes, but they have the same genes located on them. 3 and 4 and so on and so forth. And there are 23 pairs, including a pair of sex chromosomes. So half the chromosomes come from one parent, half come from the other. So this is thing that you're looking at right here is called a karyotype. It's when we can actually pull the chromosomes out of a cell that's going through mitosis and look at them and pair them up and say, oh, this is this one, this one's this one, and so on and so forth. We can learn a lot from karyotypes, and we'll talk more about this a little bit later. You guys should remember this, the detailed structure of chromosomes when they're getting ready to divide. We have our centromere that holds them together. Okay, we have where genes are located. Notice on this copy of the chromosome and this copy of the chromosome, look, the genes are located in the same location, but the alleles may be different. Maybe this gene over here, they both code for eye color, but this one codes for brown eyes, and this one codes for not brown eyes. Okay, so you should remember allele is just a version of a gene. Genotype is the genes that the organism has, so it'll be 2 letters. Phenotype is the physical appearance, or a photo-pheno. Dominant traits are the ones that are stronger. Recessive traits are the ones that are not so strong. Homozygous ones, you have 2 alleles of the same trait. These are purebred or true breeding individuals. Heterozygous, or 2 different alleles for that trait. These are hybrids. So practice. So we are going to use 2 letters to represent our genotype. Okay, a capital letter to represent the dominant form and a lowercase letter to abbreviate the recessive form. Example, if P is dominant and that equals purple, we use a big P, and little p is recessive and it equals white. So the phenotype for this flower is violet because we see it's purple, and its genotype is big P, big P. The phenotype for this flower is white, and its genotype is little p, little p. This is white, which means it has 2 copies of the recessive allele. So Punnett squares, this is the standard way of working out if it's possible for offspring of 2 parents to be. So it is a helpful tool to show combinations and predict the ratios that offsprings might take. So we begin, okay, this is how we set up a Punnett square. We begin, okay, to construct a grid, and it should have 4 boxes. So 1, 2, 3, 4. And right now we're working on something called a monohybrid cross. That means it is a cross with 1 trait. Okay, next we're going to put the genotypes of one parent across the top and the other across the left-hand side. For example, let's consider a genotype of big B crossed with little b, little b. So we will put, so this is one parent's genotype. We're going to put one big B on top of this column of boxes and the other big B on the next column of boxes. Now the other parent's genotype will end up on the side. Okay, so this little b goes across from this row, and this little b ends up across from this row. Now notice that only one letter goes above each box. So one letter above this row, one letter above this row, one letter next to this row, one letter next to this row. And it doesn't matter necessarily which parent goes on the top and which parent goes on the side. Just doesn't matter. It'll end up the same either way. So the next thing you do there we go, is we are going to fill in the boxes by copying the column and the row head letters down and across to fill the empty spaces. What that means is, since we have a big B here, I'm going to go ahead and say, okay, in this column I'm going to write big B and big B. In this column, okay, I'm going to carry this down. This one will be big B and big B. Now I'm going to carry this one across. That means this box is going to have a big B from up here and a little b from right here. This box is going to have a big B from up here and a little b from over here. This box is going to have a big B from up here and a little b from over here, and so on and so forth. And so it gets carried down like that and over like that. And now these are 4 possible combinations. These are the only 4 possible combinations that exist. Okay, so now that we have learned the basics of genetics and Punnett squares, we're going to walk through some examples. So we're going to first look at parents that are heterozygous. So we have a big W and a little w, big W, little w. Okay? So we carry them down. So big W, big W, big W, big W, little w, little w, little w. So the first hint that you should probably know is that we always write the capital letters first. Even if you didn't do them first, when you rewrite it, it will always go capital letter and then lowercase letter. And we're going to obviously say that the big W is going to be for white, which is going to be dominant. Little w is going to be for recessive, which are violet. So now let's look at this. Okay, and you also want to make sure that we can tell your capital letters from your lowercase letters. Choose your letters that you're using wisely. Make sure they make sense with the trait, but also make sure that they don't look identical. So what percentage of the offspring will have violet flowers? Well, remember, any time that there is a big capital letter, it's going to show up in the phenotype. So what genotype or phenotype will this one have? Well, it's 2 big W's. Big W is white, so this guy will be white. There's a big W and a little w. Well, the big W is there, so the dominant trait's going to show up, so this flower is white. Big W, little w, this flower is white. This one has 2 little w's, so this one must be violet. So what percentage will have violet flowers? Well, only 1 out of 4. So if I make a percentage out of that, that is 25% will have violet flowers. Okay. Now red hair is dominant to blonde hair, so we're going to make a cross between a heterozygous redhead and a blonde. So redhead, heterozygous redhead, there's the heterozygous, and the blonde. So we're going to go ahead, big R, little r, little r, little r, little r. And essentially the little r's will end up coming across, so we go little r, little r, little r, little r. Okay, so now we're going to look at it and we're going to say, okay, well, what are these? What is the phenotype of each of these? Well, this one has 1 big R, 1 little r. That means this individual would be a redhead. This one has 1 big R, 1 little r. That means this one would be a redhead. This one has 2 little r's. That means it should be a blonde-haired individual. This one has 2 little r's, so it would be a blonde-haired individual. So if we ask the question, what percentage of the offspring will have red hair? Well, this guy has red hair, this guy has red hair, blonde hair, so there would be 2 individuals out of 4, and if you do a percentage, that is 50% will have red hair. Okay, so we're going to try some more. So in pea plants, tall pea plants, big T, are dominant over short pea plants, little t. So you're going to construct a Punnett square for a heterozygous tall pea plant and a short pea plant, okay? So what are the percentages of each of the phenotypes? So I'm going to go ahead and show you this, and I want you to tell me what are the percentages of the tall pea plants and what are the percentages for the short pea plants. Go ahead and pause it if you need to. Okay, so for the tall ones, since tall is dominant, you know anytime there is a big T, we're going to have tall. So this is a tall plant, this is a tall plant, but there's no big T here, so this is a short plant, and this is a short plant. So what are the percentages of phenotypes? That means 50%, or 2 out of 4, will be tall, and 2 out of 4 will be short. Okay, tall and short. All right, so now let's look at a homozygous dominant versus a homozygous recessive. So in humans, brown eyes, big B, is dominant to not having brown eyes. If a true breeding brown-eyed person, remember that means a homozygous dominant person, is crossed with a true breeding non-brown-eyed person, remember that means homozygous recessive, what percentage will not have brown eyes? So what percentage will show the recessive trait? Okay, first we can look at this and we can say, okay, what is this one going to be? Well, it shows it has a big B, so that means it's going to be dominant, so it must be brown eyes. Okay, this one is also dominant, so that's brown eyes. This one is also dominant, so that's brown eyes. And this one is also dominant, so that's brown eyes. So phenotypic, so it is going to be 100% brown eyes. Every single offspring will have brown eyes. Now the ratio would be 4 to 0. That means you just look at, say, okay, if I say brown eyes to not brown eyes, how many brown eyes are there? There are 4. How many not brown eyes are there? There are 0, so 4 to 0. If you do genotypic, well, they're all the same genes, right? So there's no other combination, so it's 100% big B, little b, and 4 to 0 because there's no other options, big B, little b. So a heterozygous versus a heterozygous. So pink tails are dominant over gray tails in rats. So we're going to make a cross between two heterozygous individuals. So what I'd like you to do is pause really quick and try to do that on your own. All right, so if I say there are two, big T will go here, big T here, little t across this way, little t across this way. So this guy will be two big T's, so it shows the dominant trait. So this one is pink, this one is pink, and this one is pink, and this one is, well, doesn't have a big T, so it has to be gray. So if you're looking at the phenotype, okay, that means we have 3 out of 4 individuals have pink tails, so 3 out of 4 is 75% of the individuals have pink tails, and only 1 out of 4, or 25%, have gray tails. Okay? So if we're doing the ratio, we say, oh, how many versus how many have these? So we have 3 with pink tails, so 3, and how many have gray tails? 1, so 3 to 1 ratio, tall to short. Okay? Tall, short. So we list tall first, that's the 3, short second. Now the genotypes, I have 1 big T, I have 2 big T, little t's, and 1 little t. So 1 out of 4 is big T. 2 out of 4, okay, remember 1 out of 4 is 25%. 2 out of 4 is big T, remember that's 50%. And little t, 1 out of 4 is 25%. So 1 out of 4, big T, 2 out of 4, big T, and 1 out of 4, little t. So our ratio is 1 to 2 to 1, big T to little t to little t. And I can turn those into percentages, 25% big T, 50% big T, and 25% little t. All right, I hope that makes sense, and I will see you guys tomorrow. Write down any questions you have.